Thank you for joining us. This is the ASLM special COVID-19 session number 10, a series that we have dedicated to diagnostics in the COVID-19 pandemic response. Today, we are focusing on the best practices of implementing a quality assured COVID-19 testing. My name is Patrick Mateta. I'm from ASLM, and I'll be your moderator today. As usual, this will be a 1 hour long session. We shall have one presentation followed by a question and answer segment. Please enter your questions and comments into the chat box. In the session today, we intend to touch on the quality standards and issues that have a bearing on the quality of the test result to inform better control of the pandemic. That is, issues relevant to the pre-examination, the examination, and the post-examination phases of testing. We are delighted to have these coming from the perspective of the Clinical and Laboratory Standards Institute, which is a non-profit organization that promotes the development and use of voluntary laboratory consensus standards within the healthcare community. Colleagues, allow me to introduce to you our speaker today, Mr. Patrick Mateta. Mr. Mateta is a medical laboratory scientist who has been in this field for more than 25 years, working with increasing responsibility. 15 of these years have been in the laboratory systems strengthening activities, from starting up research laboratories, fully fledged multidisciplinary laboratories, conducting clinical trials, and implementing quality management systems to levels of accreditation to international standards. Mr. Mateta is a graduate of the University of Zimbabwe, Medical Technology, Nottingham Trent University, where he gained his MBA, the University of Portsmouth for his MSc in Strategic Quality Management. He is also a certified quality auditor with the American Society for Quality. Mr. Mateta has also studied the Lean Six Sigma concepts and the principles at Villanova University. He has an in-depth knowledge and understanding of international standards, including the main ISO 15189, 15190, and related. He also has experience in auditing and mentoring skills that are vital in assisting medical laboratories to implement quality management systems. Mr. Mateta has mentored and conducted laboratory audits in more than 22 countries across the 4 continents that they are working in, and he has also facilitated workshops and trained thousands of laboratory professionals. He is currently serving as the Vice President for Global Health Partnerships at CLSI, where he oversees lab system strengthening activities in more than 4 countries. He is also a board member of the African Society for Laboratory Medicine. Mr. Mateta, over to you.
Thank you, Mr. Patrick Mateta. It's an opportune moment for me to come and talk to fellow laboratorians at such a time as we are in, first with this COVID pandemic, which has really devastated communities and countries worldwide. Let me see. Is my screen? Oh, yeah, okay, let me share it now. Okay, is everyone seeing my screen now?
We are seeing both the preview, so I think you probably need to share it the other way you did it the other time.
Oh, okay. All right, let me do that.
So are you using a second screen?
Yeah.
You probably need to go to display, swap displays.
Are you seeing it now?
Yeah, now you need to put on presentation mode. Perfect.
And you are not seeing the preview, right?
Yes, now it's okay.
Okay, thank you so much. And the structure of my presentation is I'll go through introduction of some of the important issues around the CLSI support for COVID testing. And I'll go through the planning and preparation for COVID testing, and then selection, verification process for new testing methods, as well as looking at the current testing technologies that are available, and how we can also ensure that our operators are fully trained and are competent to do the tasks, as well as addressing safety practices that protect our lab staff. And I'll give you a background or a reference document or a link to what is available at CLSI as we speak. So CLSI's role in the COVID testing support, it's very important for us to just mention to you that our mission as CLSI is really to develop lab, clinical and laboratory practices and promote their use worldwide. And part of it is making sure that these resources are available and people are able to access them and utilize them. So we also recognize the contribution of lab professionals and how it is important for them to have resources that help them to face this pandemic in a safer environment that ensures that there is not many of them are affected. This global pandemic underway is really devastating the profession, because a lot of the reports that we get from different countries indicate high infection among lab professionals. So COVID testing, I think it's important for us to understand that anything needs planning. Proper planning helps us in moving forward in any lab setup, even if you are implementing a new testing technology or a new method, or you have a new test that you have introduced. It's always important to properly plan and prepare on how that technology is implemented. As a basic, in terms of the testing technologies available, we do have molecular methods on the market, which are using the usual viral RNA real-time PCR testing, or some methods that are also now coming right now, measuring the antibodies, either IgG or IgM for COVID-19. Now, each test, we need to understand that all tests really go through a product life cycle, such as like a live organism. We know there's inception until the death. So even the test itself goes through what we call a testing life cycle. And this begins with a feasibility at the manufacturer's or at the producer of the test facility. And that also looks at the design in terms of how it's going to be designed in the testing. The development itself is outlined. And then they do a major piece of what I'm going to be talking about a while from now, which is the validation piece. So the manufacturer is mandated or is supposed to actually validate any test that they develop to make sure that it's meeting the intended purpose. So the laboratories, when we get a test, our portion starts where we now do the preliminary evaluation of a test that has been validated, especially if it's not a lab developed test or if we are not changing the intended purpose of that test. So that preliminary evaluation in the lab is supposed to be called verification. I'll talk more about what verification is later on. And then once the test has been verified, it can now be launched for use in the lab. And the maintenance piece is just to make sure that on the daily basis, as we run that test, we continually perform the necessary QC to verify that it's still working, because it may work today, but tomorrow conditions may have changed and it may not be working optimally as it's supposed to be. And then if need be, there are some tests that the lab will have to retire, especially if it's no longer serving the purpose, and or new technologies that are better have been introduced. It comes a time where a test can actually be retired. Now, it's important for us to know that implementing these new tests requires a structured process that firstly starts with the lab having a plan, preparing for that test to be implemented at the lab. Then the lab must select the best test for the laboratory and verify that it works as stated by the manufacturer. The test operators, the lab people that are working on the test, they are also trained and determined to be competent to run any of the testing that's going to be performed. And of course, we cannot overemphasize the fact that sample collection and handling and transportation of those samples must be defined and in a controlled order or manner that makes sure that the testing is going to be valid, because testing can be affected by the quality of the sample. So it's very important for us to make sure that we control the processes from the time the sample is collected up to the time that it gets to the lab, to make sure that we have a quality sample that we have, and then ongoing quality checks from the pre-analytical up to the post-analytical, or in some cases, pre-examination into post-examination. So it's also important for us to understand that currently, when we're dealing with COVID testing, the tests that we are currently using, or the tests that are on the market, they are tests that have been approved for emergency use. Now, because this pandemic is an emergency, so what that means is the tests that have been approved, they didn't go through the extensive or thorough validation process that normally follows the life cycle of a test where there is no an outbreak like the one that we have currently. So I would recommend that each one of us, if we are considering using these tests, we leverage on helpful information that's available on the website for FDA. They outline some of these tests that have received emergency use approvals. And WHO also has a list of procedures that have been approved for emergency use. So it's important for us to have this information before we even choose which one to use. At least we must have tests that appear to have been either approved by the FDA or WHO. And I know in some cases this may not be the case, but it's always a backup to make sure that you are not using a test which is not known or a test that just came from a briefcase company. Now, before beginning testing, CLSI has a document, GP36A, which is really a very useful document for the laboratory in terms of how to develop, implement, and sustain effective emergency preparedness plans. It is available for free on CLSI's website, and I encourage as many of you as can access this document to do so. It's a free resource that's available. And when it comes to selection for the laboratories that are implementing ISO 15189 standards, you know that under clause 5.5, there is a list of minimum requirements to be met with regards to selection, verification, and validation of examination procedures. it's really important, and it's only minimum requirements, but I encourage all the labs to actually go in there and make sure that they address those minimum requirements, because they form the bedrock of really a quality result that we can share with the rest of the world. So method validation versus verification, you know, the analytical and clinical performance characteristics of all tests should be established prior to use with any patient population. This is actually a requirement even in the ISO, as I mentioned, I think in the pre-analytical phases or pre-examination requirements. The ISO requires that we do either validation or verification to make sure that we determine the performance characteristics of these tests. And this is done mainly through validation and verification. And validation being confirmation through the provision of objective evidence that the requirements of a specific intended use or application have been fulfilled. Whereas validation, verification is confirmation that the assay performs or its characteristics are in agreement with what the manufacturer has said the test is supposed to be doing. But this is at your lab level. So it's very important for us to know the difference between those two, because we may, in most cases, under normal circumstances, if a test is being launched, we assume that the manufacturer has had validation and the information for validation is available. And therefore, at the lab level, we may not require to do a validation as per ISO standard. We then do a verification to just make sure that in our labs, in our setup, the test still performs as has been stated by the manufacturer. Now, when to validate or verify? This is always a question that many people ask. So how do I know whether I need to verify or to validate? And this table really gives a lot of information in terms of when. And as you can see, there is only one where one of the conditions that we have to fulfill verification. Majority of them are where we do validation. But let me begin. If an assay has been fully validated and the information is available, all we need to do is verification. We need to verify what that information from the manufacturer has said, whether it conforms to what we are getting at our lab setting. For non-standard methods and any lab-developed tests or standard methods that are used outside of their scope, or standard methods that have been modified, for example, commercial kits which may be used with a specimen type not recommended by the manufacturer, or off-label or off-license or in-house, all these kind of tests will need to be validated. And the validation is more extensive and more thorough than verification. Now, method verification is really looking at a few things that, especially since these are all qualitative methods that we are looking at for COVID, we are so interested in knowing the sensitivity of the test as well as the specificity of a test. These are what key markers that, or indicators that we can use to determine whether this test is actually suitable or not suitable. It's very important for us to understand that if a test, in any test, if a test is very sensitive, what we are saying is that test is able to detect small quantities of any analyte. And in the clinical settings, they always refer that to clinical sensitivity. And sometimes you hear people talk of diagnostic sensitivity, which is really the ability of each of these tests to detect patients who have the disease or conditions for which they are being tested. And then the other term you hear, as I said, specificity. You can hear people talk about clinical specificity or diagnostic specificity. And this really is also the ability of a test to identify patients who have not, who do not have the disease or the condition for which they are being tested. In other words, we want a test which can be able to say if someone is negative, we have a high confidence level or high confidence degree with the result if it reports that this person is negative. And if someone is positive, we also want a test that is able to tell us with probably 95% confidence that this positive result is positive. And this is important because currently, if you are someone who's reading a lot of news around the COVID-19 testing, there is a lot of debate as to which of the testing technologies are being effective and which are more sensitive than the others and the impact of that. And it's very important for us to get a test that can be both highly sensitive and highly specific so that we eliminate a lot of the false positives and false negatives. Suggested protocol for qualitative method validation. This is just a brief, which I thought I need to bring to everyone's attention in case people are struggling with this. You can find 10 non-positives and 10 non-negative samples for COVID-19, for example, and calculate specificity when you run them. So you run them using the test that you are comparing with the other. So you run, they are non-positive. Let's say they are known maybe by PCR, by another reference lab. They give you those 10 samples, and then you run them using the testing method that you want to implement. And what you then do is, once you run them, you are looking at, okay, if you had 10 true positives, those true positives, were they really true positives? The most ideal would be to actually get all of them to actually be positive, just like as is in the contingents table here, and 0 negatives, because we are saying these are true positives that have been characterized by a reference lab. And then your negatives, the same, you expect the negatives to be negative, negative. And that way, if everything being equal, if you have your 10 samples that are positive and non-positive and non-false negatives, then it means your sensitivity is just these 10 divided by these 10, I mean the positive 10, that the number that you get here is true positives. Divide that by the sum of true positives and false negatives. And in this table here, it means we are saying it's 10 divided by 10. And you multiply that by 100 and to make a percentage, which means it's actually 100% sensitivity there for this test. And if it's specificity, it's also the same. You're now looking at the your true negatives. And we started with 10. And if we get 10, it means it was able to pick all the 10 as true negatives and there was no false negative. So you also then divide that the negative result by the sum of your false negative and true negatives to get your specificity. So this is just a simpler way of how we can calculate. And going ahead, it's also important for you to know that most of these tests which are on the current market have been approved by regulatory authority, as I said earlier on, for emergency use. But more so, it's then important for us as the lab to make sure that when they come to our labs, we don't just say, oh, it's approved, because we know it was approved under emergency use, which did not go through the intensive process that normally takes all the others. So it's important for us to understand that. And CLSI has a document, EP19, which is also free and describes method verification activities and gives you resources in terms of even other references that you may require in order for you to get your methods verified effectively. And then we also have, allow me to share with you some of the documents also we have at CLSI that can help you with your method verification. These are shown here. I won't go over them because you get these slides, but it's important to understand that these documents, they help you as resources that you can refer to make sure that you optimize your testing and prevent errors and ensure that every specimen that you receive is verified and the risk associated with testing is also eliminated. Whether you are new to molecular testing or very experienced, these CLSI molecular diagnostic standards will also help you through your testing using the most up-to-date protocols and procedures. In terms of quality control events, there are many quality checks that are necessary to have throughout the COVID testing process. And I will just mention that during one of the quality checks, obviously, at the beginning is the method validation or evaluation, which we talked about earlier, and then checking your instrument integrity as you continue to test before testing and even during testing, and then making sure that before your patient is tested, your system is running efficiently, and the usual day-to-day QCs that we run, and reviewing your new testing shipments. Let's say you receive new kits in your lab today, you need to make sure that those kits meet the minimum receiving requirements that you also have to set at your lab. And then making sure that every person who's working on the bench is effectively and competently trained to operate the methods or to the machines or the testing devices that you may have. So it's also very important for us to know that we need to be sure that in all these qualitative assays, we use recommended controls. Most of the kits, they come with a positive and a negative control. It's important that we do have those controls, and especially to ensure that the result as it comes, that result is usable. Otherwise, if the control fails as usual, we don't need to use that test result. Also, in the early phase of COVID, it's very important for us to understand that there are so many challenges currently, and this is expected with any new testing procedure, especially the ones that are launched as for emergency use only. Obviously, the earlier thing is there is not much performance data available for the COVID-19 test kits. And there is also a lack of positive control material for COVID method evaluation studies. And even EQA, you may actually realize that there is no valid EQA program that you can enroll into in order to ensure that your test is performing very well. So that increases the lack of confidence in ensuring true positives and true negatives. And the more reason why we need to make sure that at our lab site, we actually do the verification of any kit that we receive as we continue testing. Now, let me talk a little bit about the operator and training competence, and also refer you to the CLSI document QMS03. This guideline really provides a structured approach for developing effective lab personnel training and competence assessment program. So your staff, you need to make sure that they are trained and they are competent before they perform the testing. This is an example of a template that you can get in terms of your lab training program. It's also available in the QMS03 document that I just mentioned. Even this one as well, just showing you how you can assess your operator competence and record it and document it to make sure that it's available in the event that there is either an audit, or if you are accredited, if accrediting bodies come and they want to know your records, you can actually capture them in a worksheet like this one. And then the safety procedures also, it's very important for us to know that this disease is very infectious, and it's important to make sure that we protect our staff and we protect anyone who comes close to our facilities. So we do have documents also at CLSI GP17 and M29 that will help you to understand safety practices that need to be in place in your laboratory to protect your valuable asset, which is your personnel, and also anyone who comes in your lab environment. I'll talk briefly here about the different levels of the biosafety levels as defined by CDC. If you go on that link on the bottom of this slide, this information is available. And they have suggested, CDC has suggested that it's important that if we are doing any isolation and characterization of viral agents related to this COVID-19, we must proceed or process them in the BSL level 3. And I know this may be a challenge, but point number 2 then says if you are using BSL level number 2, the lab may perform routine diagnostic testing. And again, I encourage you to refer to that link and to get more information in terms of what can be done in BSL level 2 versus level 3. Because we don't want to just say, oh, we can do it with whatever we have, because whatever we have may actually put our staff at risk. Again, there's a ton of information that is available on the WHO website, and I encourage you also to go on that link to understand more of the procedures that we can do in terms of ensuring biosafety and the safety of our staff as they work on this disease. And additional CLSI resources and documents, there are so many information, and I think I shared with ASLM some information on what is available for free and what is also available for sale. So if the patient specimen is not good, we all know that the results that we produce are going to be a compromise. So it is important for us to make sure that the specimen identification, the collection, handling, transportation, and everything is done well to avoid contamination and anything that may compromise our test results. And then point of care tests, a lot of tests that are now coming on the market are going to be point of care tests. And it's important to understand that point of care testing in most of the settings has been performed by non-lab staff. And it's very important to actually understand that those non-lab staff need to know how it's done, and they have to also be competent. And not only competent, the safety issues that we talked about, they have to abide by those guidelines and make sure that they are safe themselves. But CLSI has a lot of documents, POCT documents, point of care testing documents, and among them there's point of care POCT 07, which is a free one. The others are for sale. And whatever you can get for free, I encourage you to get that for free. And then additionally, you can add more if you are able to purchase the additional documents. So in summary, what I've been talking about is that there is need for us to really develop a structured plan for emergency testing. Yes, COVID is here and is here to stay. What we need to do is to make sure that currently, while the disease is still unfolding, we put in place a structured plan for emergency testing. And that plan must ensure the safety of your personnel, because we don't want your personnel who are key resources to fight against this pandemic to then be victims themselves. We really want them to be strong and to remain contributing to the fight against the pandemic. And we also need to make sure that we verify all the tests that we select to use in our labs and implement your tests properly. Don't cut corners, because it will bite you. You know, there are labs right now, even here in the US, that are having questionable results because some of their procedures have not been done properly. So avoid all those things and ensure effective training. Your staff need to be competent in performing the tests that they are performing. Ensure that sample from the pre-examination phase all the way to post-examination phase, all those phases, the sample trail is actually done properly according to the guidelines that are laid either through the CDC. There are links that I have provided in this presentation for CDC guidance documents, links, and to WHO guidance documents as well, and even CLSI guidance documents. Make use of what is available and manage point of care testing. As often as we know, people think point of care is easy. It's easy, yes, if it's well managed, but if it's not well managed, it may actually be a problem. And thank you for listening, and I hope to hear questions from you. Thank you.
Thank you very much, Mr. Mateta, for such a wonderful presentation. Practical issues indeed. If I were to quote you, I think one statement really struck me: don't cut corners with quality. It will bite you. And indeed, if you do, it will bite you. And thanks for such a nice summary. We need really that structured plan. Ensure safety of our personnel is very important. Verification, we cannot never overemphasize this. I think this is a common question that keeps on coming. Verification against validation, and we don't want this to be a hindrance, especially in the emergency settings. Colleagues, we are now at a stage where we are going to be taking questions, and we are picking questions from the chat. I will pick the first question from Amha Habden Michael, and he says, how can we QA a given COVID testing lab in the absence of a gold standard? How can you quality assure a given COVID testing lab in the absence of a gold standard? What is your advice to do routine QA during this pandemic?
Okay, thank you. That's a very important question. And as I said, I think one of the slide I mentioned, the challenges of a new testing technology like this one. Obviously, EQA material is not available. So what we can do is in labs in countries where we are, obviously I assume that we have reference labs. And most of those reference labs are really our contacts in terms of getting maybe positive, non-characterized positive specimens. So at national level, probably I would prefer that the reference lab becomes the source to the eyes and the ears to monitor the other smaller labs that are testing. And if it's the only one that's testing in the country, therefore I assume they may actually work with other countries, reference labs of other countries, but it's still a challenge right now. So it has to be something that we can do at a local level to just make sure that we have something that can verify if we have non-positives, just a few. And also the challenge is we also don't want to be flying around with all these positive samples of infectious pathogens across the country. But we just have to make sure that there is something that can verify that the test we are performing is actually performing as according to expectations. So use what is available in country. If you have a reference lab, use that to maybe give you non-quantified samples of positive samples that you can use to test other sites.
Thank you, Patrick. We'll move to the next question, and a relevant one indeed. One of the key elements in assuring quality has been proficiency testing. And Samson Mucheto is asking, are there any proficiency testing providers for COVID-19?
Wow, Samson, this is an opportunity to start something. There are none at the moment. As I said, I think we were talking at this follows the previous question. So using characterized samples that are known maybe at reference lab or to maybe verify the testing performance and sharing those samples with other labs may be the only way that we can go by it now until there is a valid PT schedule or PT program that's available. Right now, as far as I know, there is none that's available.
Thank you, Patrick. I also want to add that I think as this is evolving, I think something has come up as well. The WHO, I think, have already started rolling out something in connection, I think, with the global influenza program. We initially, I think in one of the first sessions, I think QCMD were still planning to see if we can be able to get something. There are a few that are coming up, but maybe what we could do is to try and pull these and see whether they've already started running these. That's for proficiency testing. Moving on, another question from Vinny Kwanyang. Vinny says, during verification of a kit, what is the standard deviation that is acceptable from the results, sensitivity and specificity from the manufacturers?
Well, that's, to me, I would say what you need to do is, I think on the slide, one of the things that you must establish out before you run your specificity and sensitivity, you need to establish your acceptance criteria. What is acceptable at your lab? I think most of the, I would say most of the sensitivity that I have had and specificity, people accept anything that has 95% confidence interval. So in other words, I would say anything that gives you a specificity of 95% and sensitivity of 95%. But again, let's also take into account that what does that mean? What that means is if we accept a test which is 95% sensitive, 95% specific, it means there is a chance that we may miss 5% of true positives. We may miss 5% of true negatives. And that's the impact of that is huge. That's why sometimes even 95% may not be as good enough. And we may need to go for something higher. But you have to establish your own criteria at your setting. Like I said in the discussion, some results of other, some papers have already been published of other tests that are on the market that may not be performing that well. They may be even lower than 90% in terms of sensitivity and specificity, but they are being used. And the challenge is the impact in terms of the missed opportunities for the negatives and the missed opportunities for the positives. It's a situation that the lab itself has to come up with their acceptance criteria. And I would refer you also to look at CDC guidance as well as WHO guidance.
Thank you very much, Patrick. Before we move on, I also want to mention that these are relevant questions, really, indeed, and the Africa Task Force on Coronavirus will be also issuing a guidance, I think, in a couple of days or weeks to come, that will also be very beneficial in addressing these important questions that you are all asking. I also want to acknowledge Eric Opio, who said, at some point I asked about the role of ISO accreditation in all this environment that we are in as lab scientists. Now the questions are coming, and he's just appreciating the content. Thanks, Mateta. Moving on to the next question is to do with emergency use approval. And quite a number of questions were coming through here. And someone says, can you please re-explain what emergency use approval means? Don't we need to validate these kits? I think it's worth just quickly going through again.
Okay, so emergency use approval is like in an outbreak, in a pandemic like now. There are many decisions that have to be made. Do we wait until a full validation process which takes care of thousands of samples, because when the manufacturer does a validation, it's an extensive validation. They have to look at different populations. Thousands of tests are performed to make sure that this test is actually valid. In emergent cases, this is they do an abridged version of that. In other words, what I'm saying is they don't wait that long. Just like I think for some of you have been hearing a lot of discussion on the vaccine trials that need to be conducted for the development of a vaccine, and how in normal cases, a vaccine trial you cannot get a vaccine within 6 months or within a year. It has to be probably at a minimum, maybe around 18 to 24 months, because of the different phases that a vaccine has to go through in terms of testing and validation. It's the same way with the test kits. They have to validate them extensively in order to be able to put them on the market. But when we say emergency approval, we are saying it has not gone through that full cycle where it needs to be validated extensively, but there is enough evidence. They do a short, abridged form of a validation, which provides enough preliminary evidence that this can be used for, this test can actually be used for this marker. It can identify this marker. So that's what emergency use approval or emergency use license is all about. It's approved based on the preliminary findings. And I still believe that FDA or CDC or WHO will still continue to work with the manufacturer to make sure that full data is available. And when it's available, probably if it doesn't work, they may even actually be asked to withdraw it or something like that, or change something. But otherwise, it's just to address the immediate need for testing right now based on the preliminary data.
Thank you again, Patrick. And indeed, I think I remember also what you said during the presentation. This is an emergency situation equivalent to a war, but some form of guarantee of a certain level of quality is required, and that's how the emergency approval comes. And you also mentioned that do not use tests without some traceability. So a logical starting point will be those listed tests by various regulatory bodies. And more importantly, also independent evaluations are coming up. It may be important to go for those tests and then just do your simple verifications in the labs and move on quickly. Okay, I think it's also a recurring question that is talking about approvals as well. Nathan Makombe says, I would like to know the frequency of this quality control, internal and external, and the frequency of competency assessment.
Okay, so the frequency, thank you, Makombe. So the frequency of competence assessment it's really determined by the lab. As far as I know, there are people that there are labs where they say you have to be assessed for competence annually, and some say you have to be assessed for competence maybe twice a year. To me, I think what is key is to make sure that someone who's working on the bench is competent and doing what they are supposed to be doing. Then when it comes to frequency of QC, running QC, I encourage you to follow the manufacturer's instructions. And more is better, but we know it's expensive. But if the manufacturer says with every run you have to run a control, you have to just follow that instruction.
Thank you very much again, Patrick. Just to also go back to the question of PT, I see some responses in the chat. Thank you guys for responding. They are mentioning various sources, QCMD, TESIL, and Patience also says NHLS in South Africa, who have been so instrumental in providing us for many of our countries with PT samples, will also be starting soon. So that's good news to hear as well. Picking the next question, And this is from Techno Camon. I hope it's the name, not the name of the phone. He says, kindly enlighten us where BSL-3 lab is not available. Can you outline the equipments and procedures which must be available if BSL-2 lab is employed for use for COVID testing? I know you touched on this, but it's so important to at least repeat it.
Okay, so the point I was mentioning about BSL-3 is that this is what, based on the what the CDC has put as information regarding COVID lab testing and biosafety guidelines. They recommend use of biosafety level 3 if you are isolating the virus or if you are characterizing that virus. You must process them in BSL-3 lab. And if you are performing routine diagnostic tests, you can perform that under BSL-2. But again, it's important for you to make sure that you can go back and refer to the WHO, the CDC guidelines, which are on the link that's provided on the presentation. And I think it's also, there is a presentation that was given on this platform last week on biosafety. I can't remember the presenter from CDC provided information, useful information, and on how we can improve the safety of our staff as we handle this virus. So please take use and make use of the information that's available in the channels, either at CDC or WHO, or even I think ASLM has a lot of information, and these presentations that were given last week, they are still available and they can share with you the information there as well.
Thank you very much, Patrick. And indeed, we had a session on biosafety last week. Very important indeed, quite a number of questions that keep on coming, particularly relevance of BSL-2 and BSL-3. And as also Merun Dawit has mentioned that this was not clear to him. I hope it's getting clearer as we explain this. I also want to mention that I think there's an updated interim guidance from WHO dated 13th of May. For those that are on the LabCorp WhatsApp group, I think I've shared that earlier on today. And there's one thorny question around the BSL-2, whether we should be able to use it. And I think there's some clarification there that we can try to do for the near point of care. But I'll go back to the statement again that Patrick said, do not cut corners, otherwise it will bite you. So taking some more questions, interesting from Hubtown Michael again. What is the practical advice for the false negatives with respect to QA in the Sub-Saharan African context? We have had issues of false positives, false negatives coming through by virtue of the test, but then also by virtue of quality issues. Maybe you could comment on that.
So I go back to one presentation I think that Rosanna Peeling gave also on this platform. And one of the things that was suggested from a clinical point of view, we all know in the lab that most of these tests, if a test for this result is negative, in real cases, like as we were handling with HIV, that negative result, it just means at the time of testing, the antibodies were not detected. Now, is it because the antibodies were insignificant, were undetectable, or was it due to a lab error or anything? So what I think I like the kind of recommendation that was put forward before, that if any result is negative by COVID, we also don't need to disregard clinical presentation, because the doctors are managing, they are seeing the patient, and in the lab we are only seeing the specimen. And what the patient is presenting with at the clinic level may actually be suggestive of COVID, but in the lab, when we do the testing, it's negative. Now, it's important that in those cases, working together with the clinic, WHO actually recommends repeat sampling as well as testing and to do and consider multiple sample types, because a negative result may also be as a result of the sample type. So some of these tests are very sensitive based on the sample type that you use. And so it may not be picked because the sample type you used was not the appropriate or something like that. So it's important just to know that a negative result is just telling us that at this point, assuming it was correctly done, at this point of the test, you were negative. But that negative may be in the phase where you are still converting, or maybe when you have just had contact, but it's not enough to be quantified by the test method that was used. So I think let's view negative result as just at the point of the testing, not to say if you are tested negative today, then it means 2 days down the line, you still remain negative.
Thank you, Patrick. There are quite a number of questions around point of care testing. For example, Felix Adeoye, who wants, please explain further with issues of positive and negative controls in point of care testing. And I appreciate this is quite wide, but I assume maybe you would want to talk about the serology tests, in as much as we also have some point of care testing in molecular setup.
Yeah, you know, the point of care, I'm assuming you are talking of the rapid tests that we have used, for example, in HIV. The same concept that we use them then should still be applicable here. I remember what we were doing at that time, especially most of these methods that are the filtration method, where you have a positive bar which must come out, and then you say you have a positive control. We still, the recommendations for best practices are still that outside, over and above what the manufacturer gives you in the strip that you are using, you have to actually have a non-positive that you can run. You may not run it with every strip, but probably just run it frequently to make sure that that test device that you are using is actually giving you a positive result and a negative result on non-characterized samples. That way, it eliminates the reliance on the manufacturer's strip that comes embedded in there and assuming that it's positive or negative.
Thank you very much, Patrick. I think I'll take one or two more questions before we close. And I think there are just recurring questions really coming in, mostly around BSL, mostly around point of care. But there is also a question, I think, that has been recurring in the chat around the issues of Tanzania. From our perspective, we may not be able to comment on that currently because we don't have sufficient information to be able to make a fair judgment of what happened there. So it may not be proper for us to really comment on that until we get sufficient information on that. Otherwise, I think these questions are really important. To summarize what Patrick has mentioned, these are practical issues that we have faced in the lab. We are encouraged to make sure that at least there's a structured plan as we move into COVID testing. Otherwise, we will get confused along the way. Safety is really key. Verification of the selected tests that we have chosen to really use in our country. And above all, implement these tests properly by competent and effective staff, which we need to have all the time. Most importantly, as the common saying goes, garbage in, garbage out. Sample collection sample handling and transport really matters when it comes to the quality of the result, and managing point of care testing, given the dynamics that are involved, particularly being tested outside the lab. I'd like to ask Patrick to give us about 30 seconds of any of his last remarks.
Yes, thank you so much, Anaf. And to everyone in the lab, I just encourage you to stay safe. And it's better for you to actually be safe and then be able to issue a result that's reliable than just to issue a result. So it's not about issuing a result, it's about issuing a result that's a quality result and in a quality environment, and you being safe as well, because we don't want you to be a patient. We still want you to remain contributing to the fight against this pandemic. Thank you and stay safe.
Thank you, Patrick. And as usual, we will share the slides as well as the recording in a couple of days. This usually comes in as an email. It may not have attachments, but we usually put hyperlinks to the recordings and the slides in them. Just my words of closing would be just to repeat what Patrick has said. Let's not cut corners. Let's do the right thing. Until we meet again, goodbye.
Thank you. Bye-bye.